athletes that, say, win the Boston Marathon generally train at high altitudes. And that's increasing their number of red blood cells per liter of blood. So it's a little bit like blood doping without blood doping,
right?
So to speak.
So it's a legal way to increase the amount of
train under hypoxic conditions.
Hypoxia, okay. Welcome, Dr. Bill Willmore, to the podcast. Thank you very much for doing this on a short notice. I really appreciate it. I'd like to start off by asking you what got you interested in research and specifically in academia, because as I understand, you had several opportunities to get into an industry-based research, but you chose to be an academic, a very difficult path because it takes a lot of research, a lot of time and commitment to students and your own lab. So I'd like to give you the room to discuss why you chose the path you did.
Okay. Well, first of all, thanks for inviting me to this podcast.
Okay.
I really appreciate it, and gives me a chance to talk about how I basically got to where I'm at now and where I hope to go in the future, and a little bit about our work as well. So I grew up on a farm, and I was constantly surrounded with animals, and I was really interested in biology in general, and but not specifically land biology. I was interested in marine biology.
Okay.
And so I did my undergraduate at the University of Guelph, and at the time they had a marine biology program. And so it was more of a zoology program, learning about animals, the form and function of animals, how they worked. And at the time, during my time at Guelph, I was an avid scuba diver. Nice. So that's was one of my hobbies in high school, and it led into university as well. Mm-hmm. And I became interested in how animals can hold their breath for very long dives. I was also interested in something called bradycardia.
Okay. What is that? Can you explain it to us?
Bradycardia is if you were to take your face and immerse it in cold water,
okay, your
heart rate would automatically slow. And that's the nerves in your face sending signals to your heart to actually slow it down.
Interesting.
So cold temperatures can actually slow heart rate as well.
Is that a response to slow down metabolic processes?
Yeah.
Okay. What would be the advantage of that?
Well, with colder temperatures, you want to slow metabolism down a bit anyway. And those are things that I could talk about later as well.
Sure.
So when I was at Guelph, I did take biochemistry, but biochemistry at every single university is a really hard course. Mm-hmm. And I think during the time, we had a midterm and the average was failing average, and it never got any easier than that. And at the time, I didn't really see any practical application for biochemistry as well. So biochemistry didn't really interest me as I was going through. But a lot of my fourth year courses involved how organisms adapt their biochemistry to environmental extremes. Mm-hmm. So things like high temperatures, freezing cold, low oxygen, changes in salinity. And that got me thinking about stress and stress in general. And at that particular point in time, I saw both the practical and the applied applications of biochemistry. Because when you think of it, when we get invaded by a microorganism, whether it's a bacteria or a virus, that takes our bodies away from the natural state.
Right.
And this state is called homeostasis.
Okay.
And so what we try to do is we try to alter our biochemistry in order to bring our bodies back to its normal state. So I saw that this could apply to not only basic science, but also medical science as well. And so that's when biochemistry really clicked for me. And that's what kind of made my path clear.
Okay.
So every single marine biologist wants to be the next Jacques Cousteau. Do you know who Jacques Cousteau is?
I do not. I've heard of the name, but please feel free to educate me a little bit more on that.
He used to make a lot of documentary films about marine life.
Okay.
Right. And he was always in his submersibles. He was the person who developed the scuba tank.
Oh, cool. Okay.
And, he was always out at sea making these really cool documentaries about marine life. But when I found out that Jacques Cousteau was more of an engineer than a marine biologist, then marine biology became a little bit less interesting for me, and I knew I needed something else on top of that. So, and that's what got me interested in stress.
Right.
And so there's a number of different stresses. There's environmental stress, there's chemical stress, there is physiological stress, and there's cellular stress. And it was the cellular stress that I was most interested in. So I came to Carleton University and I did my PhD. My supervisor at the time was Dr. Ken Story. And I worked on turtles that could go without air for up to 4 months.
That's ridiculous.
And they do so in their natural hibernating state. These are turtles that we have around here. They're called red-eared sliders.
Okay.
They look a lot like painted turtles.
Oh, yeah. I've seen them before.
And what they do is that during the winter, they bury themselves in the bottoms of rivers and ponds. And these rivers and ponds freeze over, preventing the animal from reaching the surface and taking a breath of air. So they go the entire winter season without surfacing.
And so is the bottom of the lake is not frozen, right?
No.
So how do they not breathe at all? Or do they have any
they
gills?
don't
or
breathe.
They don't breathe at all.
Yeah. They can take
okay.
some oxygen in through what they call the cloaca, but that's rather limited in terms of the amount of oxygen that can be taken up by the turtle.
I'm guessing it's more of a diffusion type of oxygen absorption.
It is.
Okay,
Yeah.
but they're not actually breathing using their lungs.
No.
Okay.
No. So the way they survive this is that they can depress their metabolism to extremely low levels, almost pilot light levels, levels that you can almost barely detect. And this is how they spend the winter months in their natural hibernating state. So they're considered, of the vertebrates, to be the champions of what we call facultative anaerobes, or animals that can go without air for very long periods of time.
Right.
And they're fascinating. So I was studying gene expression in their brains, and because our brains cannot survive very long without oxygen. In fact, it can only survive 1 or 2 minutes without it. So we need oxygen. We're very oxygen dependent, and we have a limited, very almost non-existent ability to depress our metabolism to the same extent that turtles could.
I have a question about that. So the turtles that are able to depress their metabolism, does it depend on how large they are?
No, it doesn't depend on how large they are, but it does depend on temperature.
Okay.
So they can only really depress it at 4 degrees Celsius or lower, close to zero. And that's the temperature of the water during winter months. If you were to take that same turtle and place it in room temperature, it wouldn't be able to do the same things that it does at 4 degrees Celsius.
Do you know of key differences between humans, us, and those types of turtles that allow them to survive those low metabolic conditions? Because we can't survive that, can we?
Right. No, we can't.
Yeah. So what's the key difference between the two?
The key difference between the two is the ability to lower the metabolism to extremely low levels. When an organism goes without oxygen for long periods of time, it relies on anaerobic metabolism, or a metabolism without air. And there's certain problems that are associated with that. One of them is the buildup of acid.
Okay.
So because we're relying on a biochemical pathway called glycolysis a little bit more than if there was oxygen present, glycolysis is the only way that we can produce ATP, which is the energy currency of our cell. But if you're relying on that, you're producing a waste product called lactic acid. And lactic acid builds up and it actually drops the pH. And our blood pH is within a very narrow limit. So with the turtles, what they do is they take calcium phosphate from their shells, basically, and they use it to buffer the acid buildup that comes from anaerobic metabolism.
Interesting. Okay.
The lab also worked on frogs that could freeze solid for very long periods of time. These are, they're called spring peepers. The scientific name for the frog is Rana sylvatica, and they can freeze during the winter months, and then you can thaw them out in the spring, and they're perfectly happy.
Right.
So they actually have ice forming underneath the skin, within the veins, and they are subject to very low oxygen as well because they don't have very much blood flow moving at the time because there's ice forming. And some of the other problems that you have with ice forming outside of your cells is damage to cells due to ice crystals forming, but also when ice is forming outside of the cell, it tends to draw water from the inside of the cell to the outside.
Okay.
And so there's an osmotic pressure. There's a problem with desiccation as well, because all the water from their cells wants to move outside to form new ice. And so the way that the frogs combat that is they build up a huge amount of glucose, which they not only use as a cryoprotectant to protect against ice forming outside of your cells, but also as an osmoprotectant to keep water back in the cells at the time. And so just prior to a freeze, these frogs would spike in glucose in their cells to very high amounts. And that would protect the animal from some of the damages of ice forming inside the body.
So that amount of glucose that they're able to store, I imagine before going into that lower metabolic state, even at that low metabolism, they're able to engage it in order to kickstart the process of reanimation, I guess.
Right.
Okay.
Right.
And then how much damage is sustained or is carried over after they, what's the proper term, reawaken? What's the scientific term for that?
So if you're coming out of low oxygen, it's reoxygenation.
Reoxygenation. Okay.
And when you're coming out of freezing, it's just simply thawing.
Thawing, okay. So when they thaw, do they have significant damage as a result of ice crystals forming in and around cells?
They may have damage,
Okay.
but the key thing with these animals is that if you do sustain damage during the stress, one of the things that you could have as a backup is an excellent repair system as well. So they're able to repair the damage quite readily afterwards as well.
Okay, very interesting. So that was the research that you were doing in Dr. Ken Story's lab
Yeah,
for your PhD?
PhD.
Did you fast track as well?
It was interesting because I came out of the bachelor's and I was going into the lab as a PhD. I still had to do qualifying exams and everything that's associated with doing a PhD, but it wasn't really a fast track. It was a little bit more like BSc to PhD.
Interesting. Do those programs still exist?
Yes, they're becoming more common at other universities. So it is possible to do that. I've heard of European universities doing this a lot, and Carleton was the first place that I learned that could be done.
I was under the impression that after undergrad, you go through a master's, and then if you want to continue PhD, you can either fast track or just finish your master's and then get into a PhD program.
Yeah.
So this is even a faster way of completing a
Yeah.
PhD.
There's advantages and disadvantages to that.
Mm-hmm.
If you did a full master's and then a full PhD, you have more time to publish papers
Right.
from both degrees. Whereas if you fast track, it cuts the time down a little bit, so you have to work a bit faster in order to get the same number of publications out within the same time.
Since you're a professor, you are familiar with, having published many papers and being established here at Carleton University. I'm curious about the concept of you publish or you perish. So could you just speak on how the system has kind of, and what I mean by the system is the educational system, essentially encourages many publications in order for professors to be granted different scholarships for funding?
So I think you've hit it right on the mark. You need publications for grants, and the number of publications you have forms an assessment of the excellence of the researcher.
Mm-hmm.
To have sort of a bar which to sort of evaluate professors by as to whether they get grants or not. So publications are important for obtaining funding, for instance, from various granting agencies.
Do you think that it could be possible that with increasing quantity, the quality might be affected in some way? Because let's say professors around the world, really not just here, are encouraged to produce more and more and more papers so that they can have a better chance of getting grants approved.
Yeah, that's a big concern that's within the scientific community.
Mm-hmm.
And more so now that there are a lot of predatory journals popping up that will publish anything, basically. So that's a big concern amongst scientists. In some fields, it's easier to publish faster and to get higher quantities of publications than in others. And the journals are looked at very carefully. The type of work that's being done is looked at very carefully. So there's a lot of different evaluation criteria when you're assessing a researcher who is applying for a grant.
Okay.
From the different funding agencies.
Okay. Now to switch back to the research that you were doing, during that time while you were completing your PhD, did you already determine that you wanted to be a professor? Did you already determine that you wanted to go down the academia route, or are you kind of still thinking that, I'll figure it out as I go?
I've always wanted to go the academic route.
Okay.
I've always had that in mind. As an undergraduate, I worked at the University of Guelph. I worked at the Ontario Veterinary College, and I worked for pathologists who were actually working with marine mammals.
Mm-hmm.
And it was a little known secret of the University of Guelph at the time, but they had seals on campus. So cool. I was working at the Ontario Veterinary College with two professors who worked with the seals that we had on campus. So I would go out and help the volunteers take blood samples from the seals, and then I would take those blood samples back to the lab and isolate white blood cells, and then I was quantifying receptors on those white blood cells, aldosterone receptors, because that was important for the seals at the time. So I worked at the university every single summer as an undergraduate, and my interest in doing research never wavered from that point in time onward.
And so that kind of reaffirmed the idea that you definitely want to become an academic.
Yes.
Could you just explain what the aldosterone receptors are on the surface of this? On the surface of what, sorry?
On the surface of white blood cells.
White blood cells. Okay,
yeah. So
we don't have those.
As
humans,
do we? We could, yes. With the seals, it was important because aldosterone is a steroid hormone hormone, and it has its own receptor, and it's responsible for controlling salt intake into the cells.
Okay.
And at the time, the seals, if they get low in blood sodium, they could go into a condition called hyponatremia, which could kill the animal almost overnight.
Wow, okay.
So they had to monitor their blood salt content very carefully, and they were interested in how aldosterone was involved in controlling salt levels within the seals so that they didn't go into this hyponatremia. And so that's the research that I was involved in. So I was quantifying receptors on white blood cells for aldosterone. And I was also doing a little bit of electron microscopy on the glands that produce aldosterone, which is called the adrenal gland. It's a small organ on top of the kidney. And so I was doing some electron microscopy on seal adrenal glands.
And I imagine that was the closest thing that you got to marine biology at the time?
Yeah, at the University of Guelph was a great place to do the degree at. And we had a field trip out to the Bay of Fundy in St. John's, New Brunswick, and that was fun. But the whole program was focused on marine life, every aspect of it. So it was, I had no regrets going through that program, and it stimulated my interest in biology even more, particularly the diversity of organisms that you can find specifically in the marine environment.
Yeah.
And I was interested in how they adapt to different conditions. So that stimulated my interest in stress, which is what I work on today.
And then you ended up going into a postdoc at Boston?
Yes. I went to Brigham and Women's Hospital in Boston, Massachusetts. It's a hospital that's affiliated with Harvard Medical School. And I was working in a research lab that was in a building called the Boston Lying-In Hospital. It was where babies were delivered. But it was a building that was right across from what they called the Harvard Medical School Quad, which was the main campus of Harvard Medical School. And I worked for a hematologist, and what they were working on at the time was looking at how human cells sense a change in oxygen, right? So working on turtles was great, but I was still interested in the human condition.
Right.
And so when it came time to choose a postdoc, I knew the area that I wanted to work in, which was sensing oxygen, or how human cells sense a change in oxygen. And I had been reading all their papers, so I just wrote to all the big names in that area. And 3 of the individuals won the Nobel Prize in 2018, I think, for working out the pathway by which human cells sense a change in oxygen. And this was Doctors Peter Ratcliffe at Oxford University, Greg Semenza at the NIH in Baltimore, Maryland, and Bill Kalin, or William Kalin, who was also at Harvard. And I think he was in the Dana-Farber Hospital there.
Okay.
So I had met the individuals, and we were sort of all in this big race to find out what was the molecule that actually sensed a change in oxygen in human cells at the time. So it was fun being in Boston. I really liked the city. It's a very academic city.
How long were you there for?
I was there for 5 to 6 years.
Wow, okay.
So doing a postdoc. And it was a very interesting environment, right? If you had a question about anything at all, you could basically walk across the street and ask the world expert in that area. And so me being the curious type, that's exactly what I did.
What were some of the most things that you were curious about at the time? Who did you go ask?
Well, one of the things was an enzyme that we called the proteasome.
Okay.
And so the proteasome is basically the cell's garbage pail for a lot of different proteins that are no longer functioning. So if you need to toss a dysfunctional protein into a garbage pail, the proteasome is generally where you sent it to. And the world expert on the proteasome was just across the street. So I had a question about something about the proteasome. And so he had a big lab. His office was in the center of the lab, which was kind of an odd place to have it, but he had windows on all sides. I think it was to keep an eye on everyone at the same time.
Make sure everyone's working.
Yeah. But he invited me into his office, and he had a big bowl of popcorn outside of it. And he said, grab a handful of popcorn and come in and ask me your questions.
Nice.
So it was a very fun environment to be in, and I really enjoyed my time in Boston quite a lot.
And so when you were working on figuring out which molecule was sensing oxygen, low amounts of oxygen, what cell, you said you were working on human cells. What kind of cells were they?
There were various types of cells. So most labs work on what are called immortalized cells. And those immortalized cells can either be a cancer cell, or they can be a cell that's transformed in a way that it continuously grows and continuously divides and never fully matures.
Okay.
Right. So its cell cycle is messed up a little bit, but it continuously divides. And it provides a lot of more or less raw materials to do experiments with. And those raw materials included proteins, it included message RNA, it included DNA, and sometimes we would work with lipids and carbohydrates.
And what did you find was the signaling molecule?
So the signaling molecule was an enzyme that basically depends upon oxygen. So enzymes are protein catalysts. They take a chemical reaction and they speed it up to enormous speed, many-fold times faster. And that's because inside the proteins, they provide the site where the optimal conditions are for these chemical reactions can occur. So it actually speeds these chemical reactions up. And sometimes these chemical reactions won't even occur if the enzyme is not present. So the enzyme that turned out to be the oxygen sensor, and it was discovered by these three individuals, which I had mentioned before, Bill Carleton, Greg Semenza, and Peter Ratcliffe, turned out to be an enzyme called a hydroxylase. And this is an enzyme that utilizes oxygen to modify other proteins. So basically, when you modify a protein, you're changing its shape, and therefore you can change its function.
So a hydroxylase, correct me if I'm wrong, is a type of enzyme that adds a hydroxyl, an OH group?
Yes.
To certain regions of a protein?
Yep. Okay. Of target proteins. The proteins are made up of amino acids, and these amino acids have what are called side chains, and it's some of the side chains that get modified in this oxygen-dependent manner. So these enzymes depend on a lot of things. They depend on oxygen, of course, but they also depend on iron. They depend on a compound called alpha-ketoglutarate or oxoglutarate, and this ties it to one of the key biochemical pathways that are in the cell called the citric acid cycle. And also ascorbic acid, so vitamin C. And so that gives the importance of iron, of vitamin C, and of the citric acid cycle to sensing oxygen. But the addition of these hydroxyl groups can act like an on-off switch for the protein. So the target protein that the OH group gets added to could be degraded very rapidly, or it could affect how it interacts with other proteins. It could affect enzymatic activity. It could open or close a channel, right? It can alter the shape of the protein so that it can form a structural component of the cell. And so all of these things could be altered by just having the presence of oxygen versus the absence of oxygen.
And so then, after your research at Boston, how did you decide that you wanted to come back to Carleton, back to Ottawa?
Well, I was applying for various positions at various universities at the time and going through the interview process. When you start a postdoctoral fellowship, the key thing to do is to be starting to look towards the next step. It's important not to spend too much time being a postdoctoral fellow, because it is a sort of a comfortable lifestyle. You're only focused on research, and you don't have to focus on administration or teaching or other things.
Do you write grants during your time as a postdoc?
I did.
You did, okay.
I did. So, but they were grants for the next step.
Oh, okay.
So towards the end of my postdoc, I was writing grants for this position that I had here. So the position at Carleton in biochemistry came up, and a number of folks here were encouraging me to apply. So I applied, went through a rigorous interview process, which included a research talk and teaching a class. And then I was evaluated by the students with the same evaluation that you have at the end of the year. To evaluate your professors and how well they did on a course. And through that, I became a professor here at Carleton. So I've been here 21 years now. And during that time, I was director of biochemistry twice. Once, and then I had a little break, and then a second time. And it's been good. I've really enjoyed my time at Carleton, and the thing that I enjoy the most is meeting the students and helping them through their degrees, and then seeing where they go after that. And that's the most fun part, because we try very, very hard to get them to where they want to be after graduate school.
That's a very good point, because I think when I first met you, it was through Matt Clinch. I don't know if you're listening, Matt, but if you are, then thank you for introducing me to Dr. Willmore, because I had been looking for a professor for my undergraduate thesis, which I ended up doing with you, but it started off as a directed studies course. And at the time, I was taking fourth year biotech course with Dr. Martha Mulally, and Matt was TAing that course and then became an instructor for it. And so he had introduced me to you at the time. And yeah, I think I've been working with you ever since. It's been 2-3 years now.
Yeah. And Matt would have been a good mentor for you in particular, because I know you have an interest in biotechnology and biotechnology companies. And that was Matt's interest too. Yeah. And that's where he ended up. So he ended up with cellular, which is a company that makes therapeutic antibodies in BC. And from what I understand, he's quite happy.
Yeah, I had a chat with him a few months ago. He was really enjoying it, loving the work-life balance, loving the weather there. So I don't know. Who knows?
Especially the weather.
Especially the weather, yeah. What's to say that, though, to be honest with you, I don't particularly mind the cold. I prefer very cold weather over very hot weather.
Right.
Yeah, and I get a lot of buzz on that from family members, because they prefer having a nice, warm, tempered climate as opposed to, snow and freezing rain. So moving on to the next point, what was your first research that you started doing here at Carleton? Because you started working in hypoxia, and so how did you transition into deciding what research you wanted to work on here?
So we worked on various stresses that were involving oxygen, and there was two main ones that we were interested in. One was low oxygen, or hypoxia, and the second one was something called oxidative stress, and those two both involve oxygen. Oxidative stress is a stress that we encounter on a daily basis because our own cells produce it. And this involves the production of what are called reactive oxygen species. And they can have profound effects on all the molecules in the cell, whether it be DNA or RNA, or proteins, or carbohydrates, or lipids. All of those macromolecules can be affected by oxidative stress. And when you're looking up various diseases, if you were just to take the name of the disease that you were studying and Google it with oxidative stress, you're bound to come up with hundreds of papers that relate oxidative stress to practically any disease that's out there. So it's a very interesting topic.
So it's fully intertwined with many different pathological states, because at some level oxygen is required, and the lack of it may help proliferate certain disease, such
Right.
as cancer, for example. It's the tumor microenvironment is heavily acidic. It is very low in oxygen.
Mhm.
Can you describe the implications of how hypoxia affects cancer, or how it might potentially aggravate it?
Well, the center of particularly solid tumors are very hypoxic. The cell mass gets so dense that it's very hard for blood vessels to reach the center of solid tumors.
Mm-hmm.
And so you have a number of different conditions that exist in the center of a tumor, and this is called the tumor microenvironment. So you have low oxygen. That's one of the key things. So hypoxia is definitely associated with the tumor microenvironment. One of the other things is that you have very high oxidative stress. The number of reactive oxygen species that are produced within tumor centers is also very high. The way that the cancer combats that is that it relies on natural antioxidants, and it keeps its antioxidant levels very high in comparison to normal cells. And so if you have high oxidative stress, but you have also high protection, you can sort of balance the two. Because the cancer cells rely a lot on glycolysis, and they can run glycolysis when there's plenty of oxygen around. And this is an effect called the Warburg effect. It's essentially glycolysis out of control. And it's what a cancer cell uses to produce the energy currency of the cell. Mm-hmm. Cancer cells are rapidly dividing, and they need to duplicate their macromolecules in a very short period of time. And so you need a lot of energy to do that. And the Warburg effect allows the cancer cells to do this. The problem with runaway glycolysis is that a lot of lactic acid is produced. And so that's the third thing that you'll find in a tumor microenvironment, is that it's also very acidic because of the cellular waste product of lactic acid building up to higher levels.
ROSs or reactive oxygen species, are a great cause for cell stress.
Yes.
So I would like to expand more on the effects of cell stress and what different types of stressors there are to cells that end up, let's just keep it there.
Yeah. So reactive oxygen species themselves are produced from dimolecular oxygen. Mm-hmm. So oxygen has two atoms, oxygen atoms, which go together as O2. And when they pick up an electron, they become the first radical. So one of those oxygen molecules has an extra electron in its outer orbital. And that's a radical called superoxide. So superoxide is somewhat short-lived. It's produced by the powerhouses of our cells on a daily basis,
AKA the mitochondria.
aka the mitochondria. And a certain small percentage of the oxygen that we take in and that are used by our mitochondria, some of that, a very small percentage of it gets converted to superoxide as it picks up an extra electron. So if superoxide, which is O2, we call it O2 dot negative, picks up another electron, then the unpaired electron is now has a partner, but it also picks up two protons. It becomes hydrogen peroxide. And hydrogen peroxide is not a reactive oxygen species. It doesn't have any free electrons in any of its outer orbitals of oxygen. But it's fairly long-lived species, and it can pass through membranes. And so it can pass out of the mitochondria quite readily. And we require small amounts of hydrogen peroxide to have our cells develop. So it's actually considered now a signaling molecule, and also considered to be what we call a second messenger in many cases.
What is its exact role in terms of signaling? Why is it required for cells to develop?
For certain enzymes, they sometimes require hydrogen peroxide in terms of producing species that are essential for development. So a very low level of hydrogen peroxide is essential for cell development and for signaling. But once that builds up to a certain level, then it becomes a stress for the cell. And that's where you have oxidative stress from hydrogen peroxide.
Right.
So our mitochondria have various complexes in it, complex 1, complex 2, complex 3, and complex 4, and then ATPase. The hydrogen peroxide is produced from basically complex 1 and complex 3 of these of our mitochondrial electron transport chain. And so it's constantly being produced. And the danger of hydrogen peroxide is when it encounters, say, metals, and two metals in particular are good at catalyzing this reaction, but iron and copper. So these two metals are what we call Fenton reagents.
Okay.
And they can take hydrogen peroxide and split it in half. So on one side you have OH negative, and OH negative is pretty inert. It doesn't really react with anything. But then you have OH dot, and that's an oxygen with a free electron in its oxygen outer orbital. And so this is called the hydroxyl radical, and it will it's very reactive, and it will try to pull off both an electron and a proton from anything it comes in contact with, because it wants to become water, H2O, right? And so it's highly reactive. So if it comes in contact with a protein, it's going to pull an electron and a proton off of that protein. If it comes in contact with a lipid, it's going to do the same. And so that causes damage to those macromolecules. And in terms of proteins, it can inactivate their function. So you no longer have a functional protein. In the case of lipids, it causes something called lipid peroxidation. Do you know when fats go rancid?
Yeah.
That's a process of lipid peroxidation. So lipids come in contact with radicals. Those radicals pull protons and electrons off of them, and they themselves become radicals. And so you have this mass production in a chain reaction of radicals being produced, and that causes general breakdown of all of those macromolecules. And in terms of lipid peroxidation, that can destroy the cell because it can open up big holes in the cell, causing some of the cell deaths that we're going to talk about later on, particularly in the necroptotic ones. And so it's very, very damaging to cells. And that's why it's associated with a lot of different diseases. When these hydroxyl radicals come in contact with DNA, they can modify DNA bases, so the rungs in the ladder, so to speak, right? It can modify those and cause mutations within that. It can cause single-strand breaks in DNA, double-strand breaks in DNA. The double-stranded breaks are harder to repair, so they're quite dangerous.
So then, out of curiosity, for the fats that go rancid in the fridge, for example, where are these radicals coming from?
That's a good question. Some enzymatic reactions can create these radicals, and so if the fats go rancid basically due to the presence of oxygen
in the air.
Most likely that's where they're getting reactive oxygen species from, but they have to have a catalyst as well. And if there's any sort of trace metals that are mixed with the fats, that could cause lipid peroxidation. Any of the enzymes that generally create radicals, that could cause lipid peroxidation as well. So there could be any number of different things, but usually it's a contaminant which is producing reactive oxygen species that has gotten into the fat somehow.
And that sounds like it's almost unavoidable.
Yeah, it sets up a chain reaction, which is, there's no end to it. So radicals will create more radicals, and then you'll have general breakdown of all the lipids within that particular sample.
What other types of stress inducers are present in cells?
Well, when I was talking about reactive oxygen species, there are also chemicals that can produce that as well. So, for example, paraquat, which is a chemical that's included in some herbicides and some pesticides that are still commonly used today. It's a very potent peroxide generator, and some of the radicals that have been associated with paraquat production have also been linked to neurodegenerative diseases like Alzheimer's. Often these are associated with some neurodegenerative diseases, particularly Parkinson's disease has been linked to paraquat usage.
So what's the implication of paraquat in?
Because it's a very potent radical generator.
Okay, and then how do those affect the Parkinson's condition?
Well, radicals can also damage proteins, and when proteins get damaged, they tend to unfold. And so when they unfold, they tend to aggregate into collections of unfolded proteins. And there's some well-known unfolded proteins that are associated with various neurodegenerative diseases. And these will eventually form possibly plaques. And these plaques are great sites for the generation of these reactive oxygen species to damage normal tissue that's outside of these plaques of aggregated proteins.
Which kind of sets up a cycle in and of its own.
Yes.
Making the situation even worse. Okay. So in terms of cell stressors, you were talking about proteins that have become misfolded, or that as a result of aggregation, they no longer hold their relatively normal function, because as I understand, the function of a protein not only comes from its amino acid sequence, but also from its shape.
Right.
So there are also different types of proteins within compartments in the cell, such as the ER, the endoplasmic reticulum, that tend to modulate protein folding. Could you speak on those, and how do they become affected negatively or positively upon inducing ER stress?
Right. So the endoplasmic reticulum is an organelle. It's a series of membranes that you find within the cell, and they surround the nucleus. So if you're looking at a cell and you'll see the nucleus in the center, you'll see a series of membranes that are surrounding it, and generally this is the endoplasmic reticulum, or the ER.
And there's also a rough ER and a smooth ER.
That is correct.
Could you also explain the difference between the two?
Right. So the rough ER has ribosomes associated with it. And ribosomes are the protein manufacturing machinery of the cell. And so rough endoplasmic reticulum is responsible for making proteins that are either going to be incorporated into the outer membrane of the cell, or are going to be secreted from the cell altogether, entirely. And so this is something that's called the secretory pathway, and it starts at the end of rough endoplasmic reticulum. Smooth endoplasmic reticulum is ER that doesn't have ribosomes associated with it. And its main function is to produce lipids that create new membranes. So rough ER is responsible for making secretory proteins. Smooth ER is responsible for making lipids.
Okay. And then what are some of the known proteins within the ER that help with folding responses, or that help with folding of proteins, and that help with regulating ER stress?
So there are proteins that fold other proteins, and these are known as chaperones. If you think of a chaperone who is chaperoning kids at a party, or someone on a date, you need a chaperone, and they fold proteins within the ER as they're being made. So as the protein is coming off of the ribosome being created, it's being folded. And there's one particular protein in the ER called BIP, and it's responsible for folding a lot of these proteins that come off the ribosome into the endoplasmic reticulum as it's being folded. If something goes wrong with this process, then you have the buildup of a lot of unfolded proteins. And the way a protein folds is that it has regions within it that don't like water, and regions within it that do like water. So the protein will fold so that the regions that don't like water are all on the inside, and the regions that like water are all on the outside. If you have an unfolded protein, those regions that don't like water are now on the outside. And they're going to try and find other regions that don't like water, and that includes the same regions in other proteins. So what happens is that unfolded proteins start to aggregate, and they start even to form fibers sometimes. And once they build up to a certain point, this causes the cell stress. And this is what's known as ER stress, or endoplasmic reticulum stress, and is the buildup of these unfolded proteins. Now the cell has a response in order to break these down. So generally what happens is that if it detects an aggregate of proteins getting together, it'll try to build a membrane around it. And this membrane is called a vacuole. And so it builds a membrane around these aggregated proteins, and it's a spherical membrane that completely encases it, and it pushes it towards another organelle in the cell, which is responsible for breaking down proteins. And this organelle is called the lysosome. So these spherical membranes are called vacuoles. And so the vacuoles get pushed towards lysosomes, they fuse with them, and then all of the enzymes and machinery that's inside the lysosome, which breaks down unfolded proteins, gets a chance to break up the aggregates, break the proteins down to individual amino acids, and then the lysosome has special amino acid transporters, so it kicks all the building blocks of proteins out so that they can be used to make other proteins that are functional. If the cell fails to do that, let's say the aggregates build up to such a high extent that the cell can't handle that anymore, then it will trigger one of the cell deaths, which we'll talk about later on, which is called apoptosis.
Mm-hmm.
So when we were talking about ER stress, one of the things that can cause the unfolding of proteins is actually oxidative stress. So a lot of these stresses are found together in the same environment, and these are environments we call microenvironments. So oxidative stress will cause the unfolding of proteins, which leads to ER stress.
Mm-hmm.
Hypoxia often causes the unfolding of proteins, which leads to ER stress. So there's a lot of crosstalk between stresses, which we'll talk about in the next session. And I can go into a little bit about how those things crosstalk.
Mm-hmm. And then just one more thing to touch upon before the end of the episode. I'd like to ask about HIF-1 alpha, which is a hypoxia-inducible factor. So how does this play a role in, after hypoxia has been induced, low oxygen environment? How does this protein get upregulated? What are its main functions? And how does it affect the physiology of the cell?
Right. So this is a protein that was discovered by one of the 3 Nobel Prize winners that I was mentioning beforehand, Dr. Greg Semenza, from the NIH in Baltimore, Maryland.
Mm-hmm.
And so he discovered a transcription factor, which is a protein which binds to DNA and controls the expression of genes. And in particular, this one controlled the genes expressed under low oxygen. And he entitled it hypoxia-inducible factor. Now there's 3 of them. There's hypoxia-inducible factor 1, hypoxia-inducible factor 2, and hypoxia-inducible factor 3. And they have 2 parts to them. They have an alpha part and a beta part. And these 2 have to come together to form a functioning unit. So often the beta part, or the beta protein, is called HIF-1 beta. It has other names as well. So it's also called ARNT, or ARNT, which stands for the Arrow Hydrocarbon Receptor Nuclear Translocator. So that's why we call it ARNT.
Okay.
And ARNT just kind of hangs out in the nucleus waiting for HIF alpha to come in, partner with it, and then it binds to DNA and it activates the expression of genes that are induced by low oxygen. So that's what hypoxia-inducible factor is. And the genes that it controls seem to be involved primarily in shifting metabolism from biochemical pathways that are reliant on oxygen to biochemical pathways that don't rely on oxygen. And I had mentioned one before, which is glycolysis. Mm-hmm. So a lot of the glycolytic enzymes, the stages of that pathway, are induced by hypoxia-inducible factor. The other thing that it tries to do is tries to increase blood flow to tissues. So if you're climbing Mount Everest, you can't do it all in one shot. You have to climb up to a certain base camp, your body has to acclimate to the low oxygen at that altitude. And once it's acclimated, you can start climbing again to the next base camp, where you have to wait and your body acclimates again to that low oxygen environment, and then you can continue on. What your body is producing at the time is genes that control or increase blood flow to tissues. So one of them is a gene called erythropoietin, which is a small protein which is produced in the liver when we're a fetus. But when we're born, that function switches to the kidney. And what erythropoietin does is it goes to the bone marrow, where all of our progenitor red blood cells are hanging out, our precursor red blood cells or baby red blood cells, and they all have an erythropoietin receptor to which that erythropoietin binds. The red blood cells mature and they get kicked out from the bone marrow to the bloodstream. And that effectively increases what's called our hematocrit, which is the number of red blood cells that we have per liter of blood. So when you're climbing Mount Everest, the number of red blood cells that you have is increasing in your blood to increase oxygen carrying capacity to tissues.
And so that's done because if there are more red blood cells, then there's a better ability for them to have that gas exchange between
Carry oxygen.
Okay, in low oxygen environments.
Yeah.
And how long is the body able to sustain these hypoxic states where all of these proteins that you mentioned are able to kind of give us a little boost in a low oxygen environment?
Well, some proteins are long lasting, so they could last potentially for days. Others have a shorter lifespan, so their turnover time is much quicker. So it really depends upon the protein that you're talking about. But athletes that, say, win the Boston Marathon generally train at high altitudes. And that's increasing their number of red blood cells per liter of blood. So it's a little bit like blood doping without blood doping,
right?
So to speak.
So it's a legal way to increase the amount of
train under hypoxic conditions.
Hypoxia, okay.
Another gene that's induced is vascular endothelial growth factor, which is called VEGF. And this is important to cancer because it binds to the cells that make up blood vessels and makes more of them. So the process of making new blood vessels is a process known as angiogenesis. And that's where small capillaries split and form new blood vessels which grow into the tissue. And this is particularly important for tumors because they want to increase the blood flow to the tumor itself, right? So that it can continue to grow and get bigger, right? So cancer cells produce this vascular endothelial growth factor, which the endothelial cells can use. And in turn, the endothelial cells produce growth factors and cytokines, which the cancer cells can use. And so it's kind of a vicious cycle where the cancer cells are providing the blood vessels with what they need to make more blood vessels in this process known as angiogenesis. And the blood vessels themselves are producing growth factors and things that the cancer cells need to divide and reproduce. And so the tumor gets bigger. And a lot of cancer therapies are trying to target VEGF, basically to starve the tumor of blood and therefore oxygen in order to cause the center of these to go necrotic and kill the tumor off. Another gene that's induced is an enzyme known as inducible nitric oxide synthase. And this is an enzyme which catalyzes reaction, but one of the final products is the gas, nitric oxide, NO. And it's actually NO dot because nitric oxide is a radical in itself. And what this does is that it causes pre-existing blood vessels to vasodilate, so they become bigger. So therefore, you can have more blood moving through them at the time. So inducible nitric oxide synthase is another gene that HIF induces. So to increase blood flow to tissues by expanding capillaries. I think it's also sometimes what hospitals use to dilate blood vessels as well, is nitric oxide.
This has been very informative. Thank you so much, Dr. Willmore. I know that we're running out of time, but we will carry on next episode, or part 2 of this episode, where we talk about the different tumor microenvironments which facilitate its growth and its proliferation to other parts of the body, as well as the different stress states that we have mentioned just earlier, and how they are capable, or how cancer is capable of using them in order to give it an advantage for growth and spreading. So I'm excited to talk about that as well.
Yes.
But for the time being, thank you so much for coming on the podcast. I really appreciate your time. Thank you for
inviting me, George.
Of course.
This has been great.
Anytime. Thank you everyone for listening, and have a nice day. Take care.
You too.
Thank you.
Bye.